And Hall, Textbook of Medical Physiology, John E. Hall, Michael E. Hall. Note to in- instructors. Contact your Elsevier sales representative for teaching resources, including slides and image banks by Guyton and Hall textbooks of medical physiology. 14E or request the supporting material at dash 14th edition Guyton and Hall textbook of medical physiology, John E. H. Hall PhD, Michael E. Hall MD MS. And preface. Functional organization of the human body and control of the internal environment, chapter 1, unit 1. Physiology is the science that seeks to explain the physical and chemical mechanisms that are responsible for the origin, development, and process of life. Each type of life, from the simplest virus to the largest trees of the complicated human being, has its own functional characteristics. Therefore, the vast field of physiology can be divided into viral physiology, bacterial physiology, cellular physiology, plant physiology, vertebrate physiology, mammalia physiology, human physiology, and many other subdivisions. Human physiology. The science of human physiology attempts to explain the specific characteristics of mechanisms of the human body that makes it a living being. The fact that we remain alive is the result of complex control systems. Hunger after us seek food. Hunger makes us seek food, and fear makes us seek refuge. Sensations of cold make us look for warmth. Other sources cause us to seek fellowship and reproduce. The fact that we are sensing, feeling, and acknowledging being is part of this automatic sequence of life. These special attributes allow us to exist under widely varying conditions that otherwise would make life impossible. Human physiology links the basic sciences with medicine and integrates multiple functions of the cell tissue and organs into the functions of the living human beings. The integration requires communication and coordination by a vastity of control systems that operate at every level, from the nervous and hormonal systems that coordinate functions of cells, tissues, and organs throughout the body. Thus, the coordinated functions of the human body are much more than the realize of the total function. Although the main forces this this book is on normal human physiology, we will also discuss the some extent pathophysiology, which is the study of disorder body functions of the basis for clinical medicine.
Cells are the living units of the body. The basic living unit of the body is the cell. Each tissue or organ is an aggregate of many different cells held together by intercellular supporting structure. Unit 1. Introduction. Each type of cell is specially adapted to perform one or few particular functions. For example, the red blood cell, numbering about 20 trillion in each person, transport oxygen from the lungs to the tissue. All the red blood cells are the most abundant of any single type of cell in the body. There are also trillions of additional cells of other types of that perform the functions different from those of the red blood cell. The entire body then contains about 35 to 40 trillion human cells. The many cells of the body often differ markedly from one another, but all have certain basic characteristics that are alike. For example, oxygen reacts with carbohydrate, fat, protein to release the energy required to all cells to function. Furthermore, the general chemical mechanism for changing nutrition into energy are basically the same in the all cells. All cells deliver produce of their chemical reactions into the surrounding fluids. Almost all cells also have the ability to reproduce additional cells of their own type. Fortune, Fortunately, when cells of the particular type are destroyed, the remaining cell of this type usually generate new cells until the supply is replenished. Mechanism living of the body outnumber human cells. Microorganisms living in the body outnumber human cells. In addition to human cell, trillion of the microbes inhabit the body, living on the skin and in the mouth, gut, and nose. The gastrointestinal tract, for example, normally contains a complex of and dynamic population of 400 to 1000 species of microorganisms that outnumber our human cell. Communicates of microorganisms that inhabit the body, often called the microbiota, can cause the disease, but most of the time they live in the harmony with their human host and provide vital function that are essential for survival of their host. Although the importance of the gut microbiota in the digestion of the foodstuffs is widely recognized, additional roles for the body's microbes in nutrition, immunity, and other functions are just beginning to the appreciate and they represent all intensive area of the biomedical research.
Extracellular fluid, the internal environment. About 50 to 70% of the adult human body is fluid, mainly a water solution of ions and other substances. Although most of this fluid is inside cells is called intracellular fluid. About one third is in the space outside of cells and it is called extracellular fluid. This extracellular fluid, one third extracellular fluid, is in constant motion throughout the body. It is transported rapidly in the circulating blood and then mixed between the blood and tissues fluids by diffusion through capillary walls. In the extracellular fluid are the ions and nutrients needed by the cells to maintain life. Thus, all cells live in essentially the same environment. The extracellular fluid, for this reason, the extracellular fluid is also called the inter-environmental of the body or the milieu interieur, a term used by the great 19th century French physiologist Claude de Bernard. Cells are capable of living and performing their special functions as long as the proper concentrations of oxygen, glucose, different ions, amino acids, fatty acid strands, and other constituents are available in their internal environment. Difference in extracellular and intracellular fluids, and the external fluid contains large amount of sodium chloride and carbonate ions plus bicarbonate ions plus nutrients for the cells such as oxygen, glucose, fatty acids, amino acids. It also contains carbon dioxide that is being transported from the cells to lungs to be excreted, plus other cellular waste products that are being transported to the kidneys for excretion. The extracellular fluid contains intracellular fluid contains large amount of potassium, magnesium, and phosphate ions instead of the sodium and chloride ions found in the extracellular fluid. Special mechanisms for transporting ions through the cell membrane maintain the ion concentration difference between the extracellular and intracellular fluids. These transport processes are discussed in Chapter 4. Hemostasis maintenance of a nearly constant internal environment. In 1929, the American physiologist Walter Cannon (1871 to 1845) coined the term homeostasis to describe the maintenance of nearly constant conditions in the internal environment. Essentially, all organs and tissues of the body perform functions that help maintain these relatively constant conditions. For example, the lungs provide oxygen to the extracellular fluid to replenish the oxygen used by the cells, and kidneys maintain constant ion contractions, and the gastrointestinal system provides nutrients while eliminating wastes from the body. The various ions, nutrients, waste products, and other constituents of the body are normally regarded with a range of values rather than a fixed values. For some of the body's constitutes, the range is extremely small. Variations of the blood hydrogen ions concentration, for example, are normally less than 5 nanomoles. The blood sodium concentration is tightly regulated, normally varying only a few millimoles per liter. Even with large changes in sodium intake, these variations of sodium concentration are at least 1 million times greater than for hydrogen ions. Powerful control systems exist for maintaining concentrations of sodium and hydrogen ions as well as for most of the other ions, nutrients, and substances in the body at levels that permit the cell, tissue, and organisms to perform their normal function. Despite wide environmental variations, the challenges from injury and disease. Much of the text is concerned with how each organ or tissue contributes to homeostasis. Normal body functions require integral functions of cells, tissues, organs, and multiple nerves, hormonal and local control systems that together contribute to homeostasis and good health. Homeostatic compensations in disease. Disease is often considered to be a state of disrupt homeostasis. However, even in the presence of disease, homeostatic mechanisms continue to operate and maintain vital functions through the multiple compensations. In some cases, these compensations may lead to major deviations of the body's functions from the normal range, making it difficult to distinguish the primary cause of the disease from the compensation responses. For example, diseases that impair the kidney's ability to excrete salt and water may lead to high blood pressure, which entirely helps return excretion to normal, so that a balance between intake and renal excretion can be maintained. This balance is needed to maintain life, but over long periods of time, the high blood pressure can damage various organs, including the kidneys, causing even greater increase in blood pressure and more renal damage. Thus, homeostatic compensations that ensure after injury, disease, or major environmental challenges to the body may represent trade-offs in long term, contribute to additional abnormalities of the body function. This discipline of pathophysiology seeks to explain how the various physiological processes are all altered in diseases, or injury. This chapter outlines the different functional systems of and their contributions to homeostasis. We then briefly discuss the basic theory of the body control systems that allow the functional systems to operate in support of one another. Extracellular fluid transport and mixing system, the blood circulatory system. Extracellular fluid is transported through the body in two stages. The first stage is movement of the body through the body in blood vessels. The second is the movement of fluid between the blood capillaries and the intracellular space between the tissue cells. Figure 1.1 shows the overall circulation of blood. All the blood in the circulations traverse the entire circle and average
of the circulatory system of once each minute when the body is at rest, and as many as 6 times each minute when a person is extremely active. As blood passes through blood capillaries, continual exchange of extracellular fluid occurs between the plasma protein of the blood and the intercellular fluid that fills the intercellular spaces. This process is shown in figure 1-2. The capillary walls are permeable to most molecules in the blood plasma with the exception of plasma proteins. The blood plasma, with the exception of plasma protein, which are too large to pass through the capillaries readily. Therefore, large amount of fluid and is dissolved constitutes diffuse back and forth between the blood and the tissue spaces, as shown by the arrows in figure 1-2. This process of diffusion is caused by the kinetic motion of the molecules in the plasma and the interstitial fluid. Diffusion of the fluid and dissolve constitutes through the capillary walls and the interstitial space. That is, the fluid and dissolved molecules are continuously moving and lounging in all directions of the plasma and fluid of the intercellular spaces, as well as through capillary pores. Few cells are located more than 50 micrometers from a capillary, which ensure diffusion of almost any substance from the capillary to the cell within few seconds. Thus, the extracellular fluid everywhere in the body, both that of the plasma and that of the interstitial fluid, is continuously being mixed, thereby maintaining fluid is continually being mixed, thereby maintaining homogeneity of the extracellular fluid through the body. Origin of nutrients in the extracellular fluid. Respiratory system shows the each time blood passes through the body, it also flows through the lungs. The blood picks up oxygen in alveoli, thus acquiring the oxygen needed by the cell. The membrane between the alveolar and the lumen of the pulmonary capillaries, the alveolar membrane is only 0.4 to 2 micrometers thick, and the oxygen rapidly diffuses by micromolecular motion through this membrane into the blood gastrointestinal tract. A large portion of the blood pumped by the heart also passes through the walls of the gastrointestinal tract. Here are different dissolved nutrients including carbohydrate, fatty acid and the amino acid, and absorbed from ingested food into the extracellular fluid of the blood. Liver and other organs that perform the primarily metabolic functions. Not all substance absorbed from the gastrointestinal tract can be used in their absorbed form of many of these substances to more unusable forms and other tissues of the body. Fat cells, gastrointestinal mucosa, kidneys, endocrine glands help modifying the absorbed substance or store them until they are needed. The liver also eliminates certain waste products produced in the body and toxic substances that are ingested. Musculoskeletal system. How does the musculoskeletal system contribute to homeostasis? The answer is obvious and simple. Were it not for the muscle, the body could not move to obtain the food requires for nutrition. The musculoskeletal system also provides the modality for protection against adverse surroundings, without which the entire body, along with its homeostatic mechanism, could be destroyed. Removal of metabolic end products, removal of carbon dioxide by the lungs, all the same time that blood picks up oxygen in the lungs, carbon dioxide is released from the blood into the lung alveoli. The respiratory movement of the air into and out of the lungs carries carbon dioxide to the atmosphere. Carbon dioxide is the most abundant of all the metabolic production. Kidney. Passage of the blood through the kidneys removes most of the other substances from the plasma beside carbon dioxide that are not needed by the cell. The substances include different end products of cellular metabolism, such as the urea and uric acid. They also include the excess of ions and water from the food and accumulate in the extracellular fluid. The kidney perform their function first by filtering large quantities of plasma through the glomerular capillaries into the tubules, and they are reabsorbed into the blood substance needed by the body, such as glucose, amino acid, appropriate amounts of water, and many of the ions. Most of the other substances that are not needed by the body, especially metabolic waste products such as the urea and the creatinine, are the reabsorbed poorly and passes through the renal tubules into the urine. Gastrointestinal tract. Undigested material that enters the gastrointestinal tract and the some waste product of the metabolism and eliminate in the feces. Liver. Among many functions of the liver is detoxification or removal of the indigested drug and chemicals. The liver secretes many of these waste into the bile to be eventually eliminated of the feces.
Performs in response to the sensations. Appropriate signals are then transmitted through the motor output portions portion of the nervous system to carry out one's desires. An important segment of the nervous system is called the autonomic system. It operates at an subconscious level and controls many functions of internal organs, including the level of pumping activities by the heart, movement of the gastrointestinal tract, and secretion by many of the body glands. Hormone system located in the body and endocrine glands, organs and tissue that secrete chemical substances called hormones. Hormones are transported in the extracellular fluid to the other parts of the body to help regulate cellular functions. For example, thyroid hormone increases in the rate of most chemical reactions in all cells, thus helping set the tempo of bodily activities. Insulin control glucose metabolism. Adrenocortical hormones control sodium and potassium ions and protein metabolism. The parathyroid hormone controls bone calcium and phosphate. Thus, the hormones provide a regulatory system that complements the nervous system. The nervous system controls many muscular and secretory activities of the body, whereas the hormonal system regulates many metabolic functions. The nervous and the hormonal systems normally work together in a coordinated manner to control essential all the organ system of the body. Protection of the body. Immune system. The immune system includes white blood cells, tissue cells derived from white blood cells, the thymus, lymph nodes, and lymph vessels that protect the body organ pathogens such as bacteria, virus, parasites, and fungi. The immune system provides a mechanism for the body to carry out the following: distinguish its own cells from harmful foreign cells and substances, and destroy the invader by phagocytosis or by producing sensitized lymphatic and specialized proteins that destroy or neutralize the invader. Integumentary system. The skin and its various appendages, including hair, nail gland, and other structures, cover, cushion, and protect the deeper tissue and organs of the body, and generally provide boundary between the body's internal environment and outside world. The integumentary system is also important for temperature regulation and excretion of waste, and it provides a sensory interface between the body and the internal environment.
External
environment. The skin generally comprises about 12 to 15% of the body weight. Reproduction. Although reproduction is sometimes not considered a homeostatic function, it maintains homeostasis. It helps maintain homeostasis by generating new beings to take the place of those who are dying. This may sound like a permissive usage of the term homeostasis, but it illustrates that in the final analysis, essentially all body structures and organisms to help maintain the automaticity and continuity of life. Control systems of the body. The human body has thousands of control systems. Some of the most intricate of these systems are genetically controlled systems that operate in all cells to help regulate intracellular and extracellular function. This subject is discussed on chapter 3. Many other control systems operate within the organs to regulate functions of the individual parts and organs. Others operate throughout the entire body to control the interrelationships between the organs. For example, the respiratory system, operating in association with the nervous system, regulates the concentration of carbon dioxide in the extracellular fluid. The liver and pancreas control glucose concentration in extracellular fluid, and the kidneys regulate concentration in hydrogen, sodium, potassium, phosphate, and other ions in the extracellular fluid. Examples of control mechanisms: regulations of hydrogen and carbon dioxide oxygen concentration in the extracellular fluid. Because oxygen is one of the major substances required for chemical reactions in cells, the body has special control mechanisms to maintain at almost exact and constant oxygen concentrations in the extracellular fluid. This mechanism depends principally on the chemical characteristics of hemoglobin, which is present in red blood cells. Hemoglobin combines with oxygen as the blood pressure passes through the lungs. Then, as the blood pressure through the tissue capillaries, hemoglobin because in the capillaries. Hemoglobin, because of its own chemical efficiency of oxygen, does not release oxygen to the tissue fluid if too much oxygen is already there. However, if oxygen concentration in the tissue fluid is too low, sufficient oxygen is released to re-establish an adequate concentration. Thus, regulation of oxygen concentration is the tissues really relies on a great extent to the chemical characteristics of hemoglobin. The regulation is called the oxygen buffering functions of hemoglobin. Carbon dioxide concentration in the extracellular fluid is regulated in a much different way. Carbon dioxide is a major end production of the oxidative reactions in cells. If all the carbon dioxide formed in the cells continue to accumulate in the tissue fluids, all energy giving reactions of the cell would cease. However, a higher than normal carbon dioxide concentration in the blood excites the respiratory center, causing a person to breathe rapidly and deeply. This deep and rapid breathing increases expiration of carbon dioxide, therefore removes excess carbon dioxide from the blood tissue and fluids. This process continues until the concentration returns to normal.
Negative feedback control of the arterial pressure by the arterial baroreceptors. Signals from the sensor bioreceptor are sent to the medulla of the brain, where they are compared with the reference send point. When arterial pressure increases above normal, this abnormal pressure increases nerve impulses from the baroreceptor to the medulla to the brain, where the input signals are compared with the set point, generating an error signals that leads to decreased sympathetic nervous system activity. Decreased sympathetic activity causes dilation of blood vessels and reduced pumping activity of the heart, which return arterial pressure toward normal. Regulation of arterial blood pressure. Several systems contribute to the arterial blood pressure regulation. One of these, the baroreceptor system, is the extracellular example of the rapidly acting control mechanism. In the walls of the bifurcation region of the carotid arteries in the neck, also in the arch of the aorta in the thorax, are many nerve receptors called the baroreceptor. They are stimulated by the stress of the arterial wall. When arterial pressure rises too high, the baroreceptors send barriers of nerve impulses to the medulla of the brain. Here, these impulses inhibit the vasoreceptor centers, which in turn decrease the number of the impulses transmitted from the vasomotor center through the sympathetic nervous system to the heart and blood vessel. Lack of this impulse causes the diminished pumping activity to the heart and dilatation of the peripheral blood vessel, allowing the increased blood flow through the vessels. Both these effects decrease the arterial pressure, moving the back toward normal. Conversely, a decrease in the arterial pressure below normal relaxes the stress receptors, allowing the vasomotor center to become more active than usual, thereby causing the vasoconstriction and increased heart pumping. The initial decrease in the arterial pressure, thus initiating negative feedback mechanism that raises the arterial pressure back toward normal. Normal ranges and physical characteristics of important extracellular fluid constituents. List some important constituents and physical characteristics of the extracellular fluid along with their normal values, normal ranges, and the maximum limits without causing death. Note the narrowness of the normal range for each one. Values outside these ranges are often caused by illness, injury, and major environmental challenges. Important constituent of physical characteristics of the extracellular fluid.
Oxygen, normal value 40, carbon dioxide 45, sodium ion 142, potassium ion 4.2, calcium ion 1.2, chlorine ion 106, bicarbonate ion 24, glucose 90 by body temperature 98.4, acid base 7.4. Most important are the limits beyond which abnormalities can cause death. For example, an increase in the body temperature of only 11 degrees Fahrenheit above normal can lead to various cycles of increased cellular metabolism that destroys the cells. Note also that narrow range of acid base balance in the body with a normal pH value of 7.4 and lethal values only about 1.5 on either sides of on either sides of normal. Whenever the potassium ion is concentration decreases to less than one third normal, paralysis may result from the inability of the nerves to carry signals. Alternatively, if potassium ion concentration increases to 2 or more times normal, the heart muscle is likely to be severely depressed. Also, then the calcium ion concentration falls below about one half normal. A person is likely to experience tetanic concentration of muscles throughout the body because of the spontaneous generation of excess nerve impulses in peripheral nerves. When the glucose concentration falls between one half normally, a person frequently exhibits extreme mental irritability and sometimes even cause convulsions. These examples should be one of the apparitions for the necessity of the vast number of control systems that keep the body operating in health. In the absence of any one of the controls, serious body malfunctions or death can result. Characteristics of control systems.
The aforementioned example.
Figure 1.4, recovery or heart pumping caused by negative feedback. Figure 1 liter of blood is removed from the circulation. Death is caused by positive feedback when 2 liters or more blood is removed. For control of arterial pressure is -50 divided by +25 or -2, that is, disturbance that increases or decreases the arterial pressure does only one third as much as would occur in this control system were not present. The gains of some other physiological control system are much greater than one of the baroreceptor system. For example, the gain of the system controlling ordinary cold weather is about -33. Therefore, one can see that the temperature control system is much more effective than baroreceptor pressure control system. Positive feedback may cause a vicious cycle and death. Positive feedback may cause vicious cycle and death. Why do most control systems of the body operate by negative feedback rather than by positive feedback? If one considers the nature of positive feedback, it is obvious that positive feedback leads to instability rather than stability, and in some cases can cause death. Shows an example in which death can ensue from positive feedback. This figure depicts the pumping effectiveness of heart, showing the heart of a healthy human pumping is about Positive feedback is sometimes known as vicious cycle, but mild degree of positive feedback can overcome by the negative feedback control mechanisms of the body, and the vicious cycle then develop fails to develop. For example, if the person in the aforementioned example bleeds only 1 liter instead of 2 liters, the normal negative feedback mechanism for controlling cardiac output and arterial pressure can counterbalance the positive feedback. And the person can recover, as shown by the dashed curve of figure 1 to 4. Positive feedback can sometimes be useful. The body sometimes uses positive feedback to its advantage. Blood clotting is one of the examples of a valuable use of positive feedback. When a blood vessel is ruptured and clot begins to form, multiple enzymes called clotting factors are activated within the clot. Some of these enzymes act on other inactivates enzymes of the immediate adjacent blood, thus causing more blood clotting. This process continues until the hole in the vessel is plugged and bleeding no longer occurs. On occasions, the mechanism can get out of hand and cause formations of unwanted clots. In fact, this is what initiates most acute heart attacks, which can be caused by a clot beginning on the outside surface of an atherosclerotic plug in a coronary artery and then growing until the artery is blocked. Childbirth is another situation in which positive feedback is valuable. When urine contractions become strong enough for the baby's head to begin pushing through the cervix, stretching of the cervix sends signals to the urine muscles back in the body of the uterus, causing even more powerful contractions. Thus, the uterine contractions stretch the cervix. The cervical stretch causes strong contractions. When this process becomes powerful enough, the baby is born. If they are not powerful enough, the contractions usually die out, and a few days pass before they begin again. Another important use of positive feedback is for the generation of nerve signals. Stimulation of the membrane of a nerve fiber causes slight leakage of sodium ions through sodium channels in the nerve membrane to the fiber's interior. The sodium ions entering the fiber then change the membrane potential, which in turn causes more opening of channels, more change of potential, still more opening of channels, and so forth. Thus, a slight leak becomes an explosion of sodium entering the interior of the nerve fiber, which creates the nerve action potential. In this action potential in turn causes electric currents to flow along the outside and inside of the fiber and initiates additional action potential. This process continues until the nerve signal goes all the way to the end of the fiber. In each case in which positive feedback is useful, the positive feedback is part of an overall negative feedback process. For example, in the case of blood clotting, the positive feedback clotting process is a negative feedback process for the maintenance of normal blood volume. Also, the positive feedback that causes nerve signals allows the nerves to participate in thousands of negative feedback nervous control systems. More complex types of control systems, feed forward and adaptive control. Later in this text, when we study the nervous system, we shall see that this system contains great number of interconnected control mechanisms. Some are simply feedback systems similar to those already discussed. May are not. For example, some movements of the body occur so rapidly that there is not enough time for nerve signals to travel from the peripheral parts of the body all the way to the brain and then back to the peripheral again to control the movement. Therefore, the brain uses a mechanism called feed forward control. It causes required muscle contractions. Sensory nerve signals from the moving parts apprise the brain about whether the movement is performed correctly. If not, the brain corrects the feed forward signals that it sends to the muscles the next time the movement is required. Then, if it further occurs is necessary, this process will be performed again for subsequent movements. This process is called adaptive control. Adaptive control, in a sense, is delayed negative feedback. Thus, one can see how complex the feedback control system of the body can be. A person's life depends on all of them. Therefore, much of this text is devoted to discussing these life-giving mechanisms. Physiological variability. Also, same physiological variability such as plasma contractions to potassium, calcium, and hydrogen ions are tightly regulated. Others, such as body weight and adiposity, shown wide variations among different individuals and even in the same individual at different stages of life. Blood pressure, cardiac pumping, metabolic rate, nervous system activity, hormones, and other physiological variables change throughout the days as we move about and engage in normal daily activities. Therefore, when we discuss normal values, it is with the understanding that many of the body control systems are constantly reacting to perturbations and other variables may different individuals depending on body weight, height, diet, age, sex, environment, genetics, and other factors. Although some physiological variabilities such as plasma contraction of potassium, calcium, and hydrogen ions are tightly regulated, others such as body weight and adiposity show wide variations among different individuals and even in the same individual at different stages of life. Blood pressure, cardiac pumping, metabolic rate, nervous system activity, hormones, and other physiological variables change throughout the day as we move and about and engage in normal daily activities. Therefore, when we discuss normal values, it is with the understanding that many of the body's control systems are constantly reacting to perturbations and the variability may exist among different individuals depending on body weight and height, diet, age, sex, environment, genetics, and other factors. For simplicity, discussion of physiological functions of and focus on the average 60 kg young lean male. However, the American male no longer weighs an average of 60 kg. He weighs now 88 kg, and the average American female weighs about 76 kg, more than the average man in the 1960s. Body weight has also increased substantially in most other industrialized countries during the past 40 to 50 years. Except for reproductive and hormonal function, many other physiological functions and normal values are often discussed in terms of male physiology. However, there are clearly differences in male and female physiology beyond the obvious difference that relate to reproduction. These differences can have important consequences for understanding normal physiology as well as for treatment of disease. Age-related and other or racial differences in physiology also have important influence on body consumption, physiological control system, and pathology of disease. For example, in a lean young male, the total body water is about 70% body weight. As a person grows and ages, this percentage gradually decreases, partially because aging is usually associated with declining skeletal muscle mass and increasing fat mass. Aging may also cause a decline in the function and effectiveness of some organs and physiological control systems. These sources of physiological variability, sex differences, aging, ethnic, and racial are complex but important considerations when discussing normal physiology and the pathophysiology of diseases. Summary: for understanding normal physiology as well as the treatment of disease, the main purpose of this chapter has been to discuss briefly the overall organization of the body and the meanings whereby the different parts of the body operate in harmony. To summarize, the body is actually a social order of about 35 to 14 trillion cells organized into different functional structures, some of which are called organs. Each functional activity contributes its share to the maintenance of homeostasis in the extracellular fluid, which is called the internal movement, as well as normal conditions are mentioned in this internal environment. The cells of the body continue to live and function properly. Each cell benefits from homeostasis, and in turn each cell contributes its share towards the maintenance of homeostasis. This reciprocal interplay provides continuous automaticity of the body until one or more functional systems loses their ability to contribute their share of function. When this happens, all the cells of the body suffer extreme dysfunction leads to death. Moderate dysfunction leads to sickness. Lipids are several types of substances that are grouped together because of their common property of being soluble in fat solvents. Especially important lipids are phospholipid and cholesterol, which together constitute about 2% of the total cell mass. Phospholipids and cholesterol are mainly insoluble in water and therefore are used to form the cell membrane and intracellular membrane barriers that separate the different cell compartments. In addition to phospholipids and cholesterol, some cells contain large quantities of triglycerides, also called natural fats. In fat cells, triglycerides often account for much as 95% of the cell mass. The fat stored in these cells represent the body's main storehouse of energy-giving nutrients and can later be used to provide energy whenever it is needed in the body.
Carbohydrate play a major
Carbohydrate plays a major role in cell nutrition, and as parts of glucoprotein molecules have structural functions. Most human cells do not maintain large store of carbohydrate, and amount usually averages only about 1% of their total mass, but increases to as much as 3% of muscle cells and occasionally to 6% of liver cells. However, carbohydrate in the form of dissolved glucose is always present in the surrounding extracellular fluid. It is readily available to the cell, also a small that it is readily available to the cell. Also, a small amount of carbohydrate is stored in the cells as glycogen, an insoluble polymer, and glucose that can be depolarized and used rapidly to supply the cell's energy needs. Cell structure. The cell contains highly organized physical structures called intracellular organelles, which are critical for cell function. For example, without one of the organelles, the mitochondria, more than 95% of the cell's energy release from nutrients would cease immediately. The most important organelles and other structures of the cells are shown figure 2.2. Membranous structure of the cell. Most organelles of the cells are covered by membranes composed primarily of lipids and proteins. These membranes include the cell membrane, nucleic membrane, membrane of the endoplasmic reticulum, and membranes of mitochondria, lysosome, and Golgi apparatus.
Membrane showing that it's composed mainly of a lipid bilayer of phospholipid molecule part with large number of protein molecules producing through the layer. Also, carbohydrate molecules are attached to the protein molecules to the outside of the membrane and additional protein molecules on the inside. The lipid in membranes provide a barrier that impedes movement of the water or soluble substance from one cell component to another because molecules often penetrate the way through the membrane, thus providing a specialized pathway often organized the actual pores for passage at specific. Also, many other membrane proteins are enzymes which catalyze a molecule of different chemical reaction discussed here in the subsequent chapters. Also, many other membrane protein enzymes which catalyze a multiple of the different chemical reaction discussed here in subsequent chapters. Cell membrane. Cell membrane, also called the plasma membrane, envelops the cell and this is a thin, permeable, elastic structure. Only 7.5% of 10 nanometers thick. It is composed almost entirely of proteins and lipids. Their mixed composition is 55% proteins, 25% phospholipid, 13% cholesterol, 4% other lipids, and 3% carbohydrates. The cell membrane lipid barrier impedes protein penetration by the water-soluble substance. Figure 2.3 shows the structure of the cell membrane. Its basic structure is a lipid bilayer, which is a thin double-layered film of lipids, each layer only one molecule thick, that is continuous over the entire cell structure. Interspersed in this lipid film are large globular proteins. Basic lipid bilayer is composed of three main types of lipids: phospholipids, sphingolipids, and cholesterol. Phospholipids are the most abundant cell membrane lipids. One of each phospholipid molecule is hydrophilic, soluble in water. The other end of the hydrophobic and soluble only in fats. The phospholipid end of the phospholipids too is hydrophilic, and the fatty end protein is hydrophobic. Because the hydrophobic proteins of the phospholipid molecules are repelled by water but are mutually attracted to one another, they have a natural tendency to attach to one another in the middle of the membrane as shown in figure 2.3. The hydrophilic phospholipid proteins then constitutes the true surface of the complete cell membrane. In the contact with the intercellular water on the inside of the membrane and extracellular water on the outside surface. Lipid layer is the middle of the membrane is impermeable to the usual water-soluble substance such as ions, glucose, and urea. Conversely, fat-soluble substances such as oxygen, carbon dioxide, and alcohol can penetrate this portion of the membrane with ease. Sulfingolipids divided from the amino alcohol. Same also have the hydrophobic and hydrophilic groups that are present in small amounts in the membrane.
Especially nerve cells. Complex spheroglolipids or cell membranes are thought to serve several functions, including proteins from harmful environmental factors, single transmission, and adhesion sites for extracellular proteins. Cholesterol molecules in membrane are also lipids because their steroid nuclei are highly fat soluble. These molecules, in essence, are dissolved in bilayer of the molecule. In sense, are dissolved in the bilayer of the membrane. They mainly help determine the degree of permeability or impermeability of the bilayer or to water soluble constituents of the body fluids. Cholesterol controls much of the fluid of the membrane as well. Integral and peripheral cell membrane proteins. Figure 2 to 3 also shows globular masses floating in the lipid bilayer. These membrane proteins are mainly glycoproteins. There are two types of cell membrane proteins: integral proteins, which protrude all the way through the membrane, and peripheral proteins, which are which are attached only to the surface of the membrane and do not penetrate all the way through. Many of the integral proteins provide structural channels or pores through which water molecules and water soluble substances, especially ions, can diffuse between extracellular and intracellular fluids. These protein chains also have selective properties that allow preferential diffusion of some substances over other. Other integral proteins act as carrier proteins for transporting substances that otherwise would not penetrate the lipid bilayer. Sometimes these carrier proteins even transport substances in the direction opposite to their electrochemical guardians for diffusion, which is called active transport. Still, other acts as enzymes. Integral membrane proteins can also serve as receptors for water soluble chemicals such as peptide hormones that do not easily penetrate the cell membrane. Interaction of cell membrane receptors with specific ligands that bind to the protein sense receptor causes conformation changes in the receptor protein. This process in turn enz- enzymatically enzymatically activates the intracellular part of the protein or includes interactions between the receptor and proteins in the cytoplasm that act as second messenger, re- relying the relying relaying the signal for the extracellular part of the receptor of the interior of the cell. In this way, interior proteins spanning the cell membrane provide a means of conveying information over the environment to the cell interior. Peripheral proteins molecules
Often,
are often attached to lit integral proteins. These peripheral proteins function almost entirely as enzymes or as controllers of transport of substances through cell membrane pores. Membrane carbohydrate the cell calyx.
membrane carbohydrates occur almost invariably in combination with proteins or lipids in the form of glycoproteins and glycolipids. In fact, most of the integral proteins are glycoproteins. About 1/10 of the membrane lipid molecules and glycoproteins. The glycoportion of these molecules almost invariably produced to the outside of the dangling outward from the cell surface. Many other carbohydrate compounds called the proteoglycans, which are mainly carbohydrates bound to small protein pores, are loosely attached to the outer surface of the cell, has a loose carbohydrate coat called the glycocalyx. The carbohydrate molecules attached to the occur surround surface to the cell. The carbohydrate molecule attached to the outer surface of the cell have several important functions. Many of them.
Carbohydrate moieties attached to the outer surface of the cell have The carbohydrate molecules attached to the outer cell surface of the cell have several important functions. Many of them have a negative electric charge, which gives most cells an overall negative surface charge that repels the negative charged objects. The glycocalyx of some cells attaches to the glycocalyx of the cells, thus attaching cells to one another. Many of the carbohydrates act as receptors for binding hormones such as insulin. Building hormones such as insulin. When bound, this combination activates attached internal proteins that in turn activates a cascade of internal enzymes. Some carbohydrate moieties enter into immune reactions as discussed in chapter 35. Cytoplasm and its organisms. The cytoplasm and is filled with minute and large dispersed particles and organelles. The jelly-like fluid proteins of the cytoplasm, in which the particles are dispersed, is called cholesterol and contains mainly dissolved proteins, electrolytes and glucose. The dispersed in the cytoplasm are natural fat globules, glucosylgranules, ribosomes, secretory vesicles, and 5 especially important organelles: the endoplasmic reticulum, the Golgi apparatus, mitochondria, lysosome, and peroxisomes. Endoplasmic reticulum shows the endoplasmic reticulum, a network of tubular structure called cisternae, that flat vascular of tubular structures called cisternae structure in the cytoplasm. These organelles helps process molecules made by the cells and transport them to their specific destinations inside or outside the cell. The tubules and vesicles interconnect. Also, their walls are constructed of lipid bilayer membranes that contain large amount of protein similar to cell membrane. The total surface area of the structure is called cells. In liver cells, for example, can be as much as 30 to 40 times the cell membrane area. The detailed structure of a small protein of endoplasmic reticulum is shown in figure 2.4. The space inside the tubular and vesicle is filled with endoplasmic matrix, a watery medium that is different from fluid in cholesterol outside the endoplasmic reticulum. Electron micrograph shows that the space inside the endoplasmic reticulum is connected with the space between the two membrane surfaces of the nuclear membrane. Substances formed in some parts of the cell enter the space of the endoplasmic reticulum and are called and then directed to the parts of the cell. Also, vast surface area of this
and multiple enzyme system attached to its membrane provide the mechanism for a major share of the cell metabolic function. Ribosome and rough granular endoplasmic reticulum attached to the outer surface of many parts of the cytoplasm. Reticulum are large number of minute granular particles called the ribosomes. Where these particles are present, the reticulum is called the rough endoplasmic reticulum. The ribosomes are composed of the mixture of RNA and proteins. They function of synthesize new protein and molecules in the cell, as discussed later in this chapter in chapter 3. Smooth a granular endoplasmic reticulum. Part of the endoplasmic reticulum has no attached ribosome. This part is called the smooth granular endoplasmic reticulum. The smooth reticulum function for the synthesis of lipid substances and for other process of the cell promoted by the intercellular enzyme. Golgi apparatus. The Golgi apparatus shown in figure 2.5 is closely related to the endoplasmic reticulum. It has membranes similar to those of smooth endoplasmic reticulum. The Golgi apparatus is usually composed of 4 or more structured layer of thin fat and closed vesicle layer near one side of the nucleus. This apparatus is prominent in secretory cells, where it is located on the site of the cell from the which secretory substances are extruded. The Golgi apparatus function is associated with the endoplasmic reticulum, as shown in figure 2.5. Small transport vesicles, also called the endoplasmic reticulum vesicles, continually pinch off from the endoplasmic reticulum and shortly thereafter fuse with the Golgi apparatus. In this way, substance interrupted in endoplasmic reticulum vesicle are transported from the endoplasmic reticulum to the Golgi apparatus. The transported substance and then proceed the Golgi apparatus of the form of lysosome, secretory vesicles, and other cytoplasm components discussed later in chapter. Lysosome. Lysosomes shown in figure 2.2 are vesicle organelles that form by the breaking of some form of Golgi apparatus. They then disappear throughout the cytoplasm. The lysosome provide an intercellular digestive system that shows the cells to digest the following damaged cellular structure: food particles that have been ingested by the cell, unwanted matter such as bacteria. Lysosomes are different in various cell types but are usually 250 to 750 nanometers in diameter. They are surrounded by typical lipid bilayer membrane and are filled with large number of small granules 5 to 8 nanometers in diameter, which are protein arranges of such many as 40 different hydrolase enzymes. A hydrolytic enzyme is capable of splitting an organic component into molecule with one part of the compound and combining hydroxyl portion of the water molecule with the other part of the compound. For example, protein is hydrolyzed to form the amino acid glycogen is hydrolyzed to form the glucose, and lipids hydrolyzed to form fatty acid and glycerol. Hydrolytic enzymes are highly considered is lysosome. Ordinarily, the membrane surrounding the lysosome prevents the enclosed hydrolytic enzyme from coming into contact with the substance in the cell and therefore prevents their digestive actions. However, some condition of the cell break the membranes of lysosome, allowing releasing of the digestive enzymes. These enzymes then split the organic substance with which they come to contact into a small, highly diffuse substance such as amino acid, glucose. Some of the specific function of the lysosome are discussed later in chapter. Peroxisomes. Peroxisomes are physically small, similar to lysosome, but they are different in two important ways. First, they are believed to be formed by the self-replication or perhaps replication. গুচ্ছগুচ্ছভাবে হয় একটার পর একটা হয় আর কি, or perhaps by budding off from the smooth endoplasmic reticulum. Budding off বাচ্চা হয় আর কি, বাডিং থোকা টোকা। তারপরে হচ্ছে rather than form of the Golgi apparatus. Second, they are contain oxidase rather than hydrolyze. Several of the oxidase are capable of combining oxygen with hydrogen derived from different intercellular chemicals to form of the hydrogen peroxide. Hydrogen peroxide is a highly oxidizing substance and is used in association with catalase, another oxidase enzyme present in large quantities in peroxisome to oxidize many substances that might otherwise be proteinaceous to the cell. For example, about half the alcohol that a person drinks is oxidified into acetaldehyde by the peroxism of the liver cell in this manner. A major function of peroxisome is the catabolize long chain fatty acid secretory vesicles. One of the important functions of many cells in secretion of the special chemical substance. Almost all such secretory substances are formed by the endoplasmic reticulum. Golgi apparatus systems are then released from the Golgi apparatus into cytoplasm in the form of storage vehicle called secretory vesicle or secretory granules. Figure 2.6 shows typical secretory vesicle inside the pancreas acinar cells. These vesicles store protein proenzymes, enzymes that are not yet activated. The proenzymes are secreted later through the outer cell membrane into the pancreatic duct and then into the duodenum when they become activated and perform digestive function on the food in the intestinal tract. Mitochondria. The mitochondria shows in figure 2.2, figure 2.7 are called the powerhouse of the cell. Without them, cells would be unable to exert enough energy from the nutrients and essentially all cellular function would cease. Mitochondria are present in all areas. A mitochondrial section, outer membrane, inner membrane, crystal, matrix, oxidative phosphatic enzyme.
But the total number of per cell varies from less than 100 to several thousand, depending on energy requirements. The cell, cardiac muscle cells, cardiocytes, for example, are large amounts of energy and have far more mitochondria. Fat cells, adipocytes, which are much less active and use less energy. Furthermore, the mitochondria are concentrated in those portions of the cell responsible for major shells of its energy metabolism. They are also variable in size and shape. Some mitochondria are only a few hundred nanometers in diameter and are globular in shape, while other are elongated and are as long as 1 micrometer in diameter and 7 micrometers long. Still others are branching and filamentous. The basic structure of mitochondria shown in figure 227 is composed mainly of two lipid bilayer protein membranes, an outer membrane and an inner membrane. Many folds of the inner membrane are cells are many membranes manifest from the cells or tubules called cristae into which oxidative enzymes are attached. The cristae provide a large surface area for chemical reactions to occur. In addition, the inner cavity of the mitochondrion is filled with a matrix that contains large quantities of dissolved enzymes necessary for extracting energy from nutrients. These enzymes operate in association with oxidative enzymes on the cristae to cause oxidation of nutrients, thereby forming carbon dioxide and water, and at the same time releasing energy. In the liberated energy is used to synthesize high energy substances called adenosine triphosphate. ATP is then transported out of the mitochondria and refused throughout the cell to release its own energy, whether it is needed for performing cellular functions. The chemical details of ATP formation by mitochondria are provided in chapter 68, but some basic infections of ATP is the cell are introduced later in the chapter. Mitochondria are self-replicative, which means that one mitochondria can form a second one, a third one, and so whenever the cell needs increased amounts of ATP. Indeed, the mitochondria contains a DNA similar to that found in cell nucleus in chapter 3. We will see the DNA is the basic constituent of nucleus that controls replications in the cell. The DNA of the mitochondria plays a similar role, controlling replications of the mitochondria on cells that are faced with increased energy demands. For example, a skeletal muscle is subject to chronic exercise training may increase the density of mitochondria to supply the additional energy required. Cell is cytoskeleton, filament, and tubular structures. The cell cytoskeleton is a network of fibril proteins organized into filaments of tubules. These originate as protein proteins synthesized by ribosomes in cytoplasm. The precursor molecules then polymerized into form filaments. As an example, large number of actin microfilaments frequently occur in outer zone of cytoplasm called the ectoplasm to form an elastic support of the cell membrane. Also, in muscle cells, actions and myosin filaments are organized into a special contractile machine that is base for muscle contraction as discussed in chapter 6. Intermediate filaments are generally strong, rope-like filaments that often work together with microtubules, providing strength and support for the fragile tubular structure. They are called intermediate because their average diameter is between that of a narrower action mitochondria and wider myosin filaments that are found in muscle cells. Their functions are mainly mechanical and they are less dynamic than actin microfilaments or microtubules.
All cells have the intermediate filaments. Have intercellular filaments, although the protein's subunits of this structure vary depending on the cell type. Specific intermediate filaments found in various cells, including Discman filaments in muscle cells, neurofilaments in the neurons, and the keratins in epithelial cells. A specialized type of stiff filaments composed of polymerized tubulin molecules is used in all cells of the constitutes a strong tubular structure. The microtubules figure 2 to 8 shows the typical microtubules of the cell. Another example of the microtubules is the tubular skeletal structure in the center of the East calcium that radiates the upward from the cytoplasm to the tip of the calcium. The structure is discussed later in the chapter. Figure 2 to 18. Also, both of the centrioles and the mitotic spheres of the cells undergoing mitosis are composed of stiff microtubules. A major function of the microtubules is to act as a cytoskeleton, providing the rigid physical structure for the certain parts of the cell. The cell cytoskeleton not only determines the part of the cell cytoskeleton not only determines cell shape but also participates in the cell division, also allows it to move and provide a track-like system that directs the movements of the organelles in the cell. Microtubules serve as a conveyor belts for the intercellular transport of the vehicles, granules, organelles such as mitochondria. Nucleus is the controlled center of the cells, sends message to the cell to grow, mature, replicate, or briefly nucleus contain large quantities of DNA. for cellular function. Energy from ATP is used to promote three major categories of cellular function: transport of substance through multiple cell membrane, synthesis of chemical compounds throughout the cell, and the mechanical work. These use of ATP are illustrated by the examples in figure 216. To supply energy for the transport of sodium through the cell membrane, to promote protein synthesis by the ribosome, and to supply the energy needed during muscle contraction. In addition to the membrane transport of sodium, energy from ATP is required for the membrane transport of potassium, calcium, magnesium, phosphate, chloride, urate, and hydrogen ions, and many other ions, as well as various organic substances. Membrane transport is as various organic substance. Membrane transport is so important to cell function that some cells, the renal tubular cell for example, use as much as 80% of the ATP that they form for this purpose alone. In addition to synthesizing protein, cells makes phospholipids, cholesterol, purines, pyrimidines, and many other substances. Synthesis of almost any chemical compound requires energy. For example, a single protein molecule might be composed of as many as several thousand. Equal might be composed of as many as several thousand amino acids attached to one another by prepaid linkage. The formation of each these linkage requires energy derived from the breakdown of four high energy bonds. Thus, many thousand ATP molecules must release their energy as much as protein molecule is formed. Indeed, some cells as much as 75% of all the ATP formed in the cell. Simply to synthesize new chemical compounds, especially protein molecules. This is particularly true during the growth phase of cells. Another use to ATP to supply energy for special cells to perform the mechanical work. We discussed in chapter 6 that each contraction of a muscle fiber requires the expenditure of large quantities of ATP energy. Other cells perform mechanical work in other ways, especially by the cell perform mechanical work in other ways, especially by the ciliary and the ambient motion. Describe the later in this chapter. The source of energy for all these types of mechanical work is ATP. In summary, ATP is readily available to release its energy rapidly where it is needed in the cell. To replace ATP used by the cell, much slower chemical reactions break down carbohydrate, fat, and proteins and use the energy derived from those processed to form new ATP. More than 95% of this ATP is formed in the mitochondria, which is why the mitochondria are called the powerhouse of the cells.
Locomotions of cells. The most obvious type of movement in the body is that which occurs in skeletal, cardiac, and smooth muscle cells, which constitute almost 50% of the entire body mass. The specialized function of these cells are discussed in chapter 6 and through 9. Two other types of movement, amoeboid locomotion and ciliary movement, occur in other cells. Amoeboid movement is a crawl-like movement of an entire cell in relation to its surroundings, such as movement of white blood cells through tissues. This type of movement gets its name from the fact that the amoeba move in this manner. The amoeba have provided an excellent tool for studying the phenomenon. Typically, amoeboid locomotion begins with the protrusion of a pseudopodium from one end to the cell. The pseudopodium projects away from the cell body and partially secures itself in a new tissue area. Then the remainder of the cell is pulled towards the pseudopodium. Figure 2 to 17 demonstrates this process, showing an elongated cell, the right hand end of which is protruding pseudospodium. The membrane of this end of the cell is continually moving forward, and the movement of the left hand and the cell is continually moving along as the cell moves. Mechanism of amoeboid locomotion. Figure 2 to 17 shows the general principle of amoeboid motion. Basically, this results from the continual formation of new cell membrane at the leading edge of pseudopodium and continual absorption of the membrane in the mid and near portion of the cell. Two other affected areas also essential for forward movement of the cell. The first is attachment of the pseudospodium to surrounding tissue so that it becomes fixed in its leading position, while the remainder of the cell body is being pulled forward towards the point of attachment. This attachment is caused by receptor proteins that line the inside of exocytic vesicles. When the vesicles become part of the pseudopodial membrane, they open so that their insides invert to the outside, and the receptors now protrude to the outside and attach to ligands in the surrounding tissues. At the opposite end of the cell, the receptors pull away from their ligands and from new androcytic vesicles. At the opposite end of the cell, the receptors pull away from their ligands and form new androcytic vesicles. Then, inside the cell, these vesicle streams toward the pseudopodal end of the cell, where they are used to form new membrane for the pseudospodium. The second essential effect for locomotion is to provide the energy required to pull the cell body in the direction of the pseudopodium. A moderate to large amount of the protein action in the cytoplasm of all cells. Much of the action in the form of single molecules that do not provide any motive power. However, these molecules polarize to form a filament network, and network contracts with its binds with an action binding protein such as myosin. The entire process is energized by high protein compound ATP. This is what occurs in the pseudospodium of moving cell, where such as network of action filaments comes a new filaments comes a new inside the enlarging pseudospodium. Contraction also occurs in electroplasm of the cell body, where pre-exciting action network is already present beneath the cell membrane. Type of cell that exhibit amoeboid locomotion. The most common cells to exhibit amoeboid locomotion in the human body are the white blood cells when they move out of the blood into the tissue to form tissue macrophage. Other types of cells can also move by amoeboid locomotion under certain circumstances. For example, fibroblasts move into a damaged area to help repair the damage, and even the germinal cells of the skin, although ordinarily completely sessile cell, move towards a cut area to repair the opening. Cell locomotion is also especially important in the development of the embryo and fetus after fertilization of an ovum. For example, embryonic cell often must migrate along distance from their site of origin to new areas during the development of special structures. Some type of cancer cells, such as sarcomas, which arise from connective tissue cells, are especially proficient at amoeboid movement. This partially accounts for the relative rapid spreading of one part of the body to another, known as metastasis. Control of amoeboid locomotion, chemostasis. An important interior of amoeboid locomotion is the process called chemotaxis, which results from the appearance of certain chemical substances in the tissue. Any chemical substances that cause chemotaxis to occur is called a chemotactic substance. Most cells that exhibit amoeboid locomotion move toward the source of a chemotactic substance, that is, form an area of lower concentration towards the area of higher concentration. This is called positive chemotaxis. Some cells move away from the source, which is called negative chemotaxis. How does chemotaxis control the direction of amoeboid locomotion? Although the answer is not certain, it is known that the site of the cell most exposed to the chemotaxis substance develop membrane changes that cause of pseudoporal protrusion, cilia, and ciliary movements.
There are two types of motile and non-motile or primary cilia. Motile cilia can undergo a wild whip-like movements on the surface of the cells. This movement occurs mainly in two places in the human body: on the surface of the respiratory areas and on the inside the surface of the urinary bladder. Uterine tubes, fallopian tubes, not urinary bladder. On the surface of the respiratory airways and the inside surface of the uterine tubes or fallopian tubes of the reproductive tract. In the nasal cavity and lower respiratory airways, the whip-like motion of motile cilia causes a layer of mucus to move at a rate of about 1 centimeter per minute toward the pharynx. In this way, the continually ciliary these passageways of muscle and particles that have become trapped into the mucus. In the uterine tube, cilia cause slow movement of the fluid from the ostium tube, cilia passageway of muscles and particles that have become trapped in the muscles. In the uterine tube, cilia cause the slow movement of fluid from the ostium of the uterine tube towards the uterine cavity. These movements of uterine tube fluid transfer to the ovum from the ovary to the uterus. As shown in figure 2 to 18, a calcium has the appearance of a sharp-pointed straight or curved hair that project of a pointed straight of curve projects of sharp pointed 2 to 4 micrometers from the surface of the cells. Often, many motile cilia projects from a single cell. For example, many motiles as many as 200 cilia on the surface of each epithelial cell inside the respiratory passageway. The calcium is covered by the outcropping of the cell membrane and is supported by 11 microtubules, 9 double tubules located around the periphery of the cilium and two single tubes down to the centers. This demonstrated in the cross section show the 2 to 18. Each cilium is an outgrowth of a structure that lies immediately beneath the cell membrane called the basal body of the cilia. The fragment flagellum of a sperm is similar to the motile cilia. In fact, it has much the same type of structure and same type of the contractile mechanism. The flagellum, however, is much longer and moves its sinusoidal waves inside the whip-like movements.
Figure 118, 2.18 Structure and function of the cilium. Modified term. In the inset of figure 2 to 18, movement of motile cilium is shown. The cilia moves forward with a sudden, rapid, whip-like stroke 10 to 20 times per second, bending sharply where it projects from the surface of the cell. Then it moves backward slowly to its initial position. The rapid forward thrusting whip-like movement pushes the fluid forward thrusting whip-like motion fluid lying adjacent to the cell in the direction of the cilium. Cell moves. The slow dragging movement in the backward direction has almost no effect on fluid movement. As a result, the fluid is continually propelled in the direction of the fast forward stroke. Because most motile ciliate cells have large number of cilia on their surface, and because all the cilia are oriented in the same direction, it is an effective means of moving forward from one part of the surface to another. Mechanism of ciliary movement. Although not all aspects of ciliary movement are known, we are aware of the following elements. First, the 9 double tubes of 2 single tubules are all linked to one another by a complex of protein across linkage. The total complex of tubules and cross linkages is called the axoneme. Second, even after removal of the membrane and destruction of other elements of the cilium in addition to the axoneme, the cilium can still beat under appropriate conditions. Third, two conditions are necessary for continued beating of the axon after removal of other structure of the cilium. 1. The availability of ATP, appropriate ionic conditions, especially appropriate concentrations of magnesium and calcium. Fourth, during forward motion of the cilium, the double tubules on the front edge of the cilium side outward towards the tip of the cilium, whereas those on the back edge remain in place. Fifth, multiple protein arms composed of the protein dynein, which has adenosine triphosphate enzyme activity, project from each double tubule toward an adjacent double tube. Given the basic information, it has been determined that the release of energy from ATP in contact with the ATPase dynein arms cause the head of these arms to crawl rapidly along the surface of the adjacent double tube. If the front tubules crawl outwards while the back tubules remain stationary, bending occurs. In the same way in which cilia contractions in control is not well understood, the cilia of some generic abnormal cells do not have the two central single tubules. These cilia fail to beat. Therefore, it is presumed that some signal, perhaps an electrochemical signal, is transmitted along these two central tubules to activate the adenine arms. Non-motile primary cilia serves as cell sensory and tannin. Primary cilia are non-motile and generally occur only as a single cilium on each cell. Although the physiological functions of a primary cilia are not fully understood, current evidence indicates that they function as cellular sensory antennae, which coordinates cellular signaling pathways involved in chemical and mechanical cessation, signal transduction, and cell growth. In the kidneys, for example, primary cilia are found in most epithelial cells of the tubules, projecting into the tubule lumen and acting as a flow sensor. In response to fluid flow over the tubular epithelial cells, the primary cilia bend and cause flow include charges in intracellular calcium signaling. These signals in turn initiate multiple effects on the cells. Defects in signaling by primary cilia in renal tubules epithelium cells are thought to contribute to various disorders, including the development of large fluid-filled cysts, a condition called polycystic kidney disease. Chapter 3: Generic control of protein synthesis, cell function, and cell reproduction. Genes, which are located in the nuclei of all cells of the body, control heredity from parents to children, as well as the daily function of all body cells. The genes control cell function by determining which structures, enzymes, and chemicals are synthesized within the cell. Figure 3.2.1 shows the general schema of genetic control. Each gene, which is composed of deoxyribonucleic acid, controls the formation of another nucleic acid, ribonucleic acid. This RNA then spreads throughout the cell to control formation of a specific protein. The entire process, from transportation of genetic code in the nuclei to translation of the RNA code and the formation of proteins in the cell cytoplasm, is often referred as a gene expression. Because the human body has approximately 20,000 to 25,000 different genes that code from protein in each cell, it is possible to form a large number of different cellular proteins. In fact, RNA molecules transcribed from the same segment of DNA, the same gene, can be processed in more than one way by the cell, giving rise to alternative versions of protein. The total number of different proteins produced by the various cell types in humans is estimated to be at least 100,000. Some of the cellular proteins are structural proteins, which, in association with various lipids and carbohydrates, form structures of various cellular organelles discussed in Chapter 2. However, most of the proteins are enzymes that catalyze different chemical reactions in the cells. For example, enzyme protein all the oxidative reactions that supply energy to the cell along the synthesis of all the cell chemicals such as lipids, glycogen, and adenosine triphosphate ATP.
Cell nucleus. Genes control protein synthesis. In the cell, molecules, large number of genes are attached end to end on end in extremely long double-stranded helical molecules of DNA having molecule weights measured in the billions. A very short segment of such a molecule is shown in figure 3.2. The molecule is composed of several simple chemical compounds bound together in a regular protein, the details of which are explained in the next few paragraphs. Building blocks of DNA. Figure 3.3 shows the basic chemical compounds involved in the formation of DNA. These compounds include the following: phosphoric acid, a sugar called deoxyribose, and four nitrogenous bases, two purines, adenine and guanine, and guanine, and two pyramids, thymine and cytosine. The phosphoric acid and the deoxyribose from the two helical strands that are backbone of the DNA molecule, and the nitrogenous bases lie between the two strands and connect them as illustrated in figure 3.2. Nucleotides. The first stage of DNA formation is to combine one molecule of phosphoric acid, one molecule of deoxyribose, and one of four bases to form an acid molecule. Four separate nucleotides and are the formed cone of each of the four bases deoxyadenylic, deoxythymidylate, deoxyguanidate, and deoxycytodialic acids. Shows as chemical structure of the deoxyadenylic acid. Figure 3.5 shows simple symbols for the nucleotides and that from DNA. Nucleotides are organized to form two strands of DNA loosely bound of each other. Figure 3.2 shows the manner in which the multiple nucleotides and bound together to form two strands of DNA. The two strands are in turn loosely bounded with each other by weak hour cross the linkage as illustrated in figure 3.6 by the central dashed line. Note the backbone of each DNA strand is composed of alternating phosphoric acid and the deoxyribose molecules. In turn, purine and pyrimidine bases are attached to the sides of the deoxyribose molecules. Then, by the means of those hydrogen bond dashed between the purine and pyrimidine bases. The two respective DNA strands are held together. Note the following carbides. However, each purine base adenine of our strand always bonds with pyrimidine base thymine of the other strand. Each purine base guanine always bonds with a pyrimidine base cytosine. Thus, in figure 3.6, the sequence of complementary pairs of bases of CG, CG, GC, TA, CG, TA, GC, AT, and AT. Because of the looseness of the hydrogen bond, and the two strands can pull apart with ease, and they do say many times during the courses of their function in the cell. To the put of DNA of figure 3.6 into its proper physical perspective, one could merely pick up the two ends and twisted them into a helix. 10 pairs of nucleotides are present in each full turn of the helix in the DNA molecules. Genetic code. The importance of DNA lies in the ability to control the formation of proteins in the cell, which it achieves by means of genetic code. This is when the two strands of DNA molecules are split apart, the purine and pyrimidine bases projecting to the side of each DNA strand are exposed as shown by the top strand in figure 3 to 7. It is these projecting bases that form the genetic code. The genetic code consists of successive triplets of bases. This is each three successive bases is code to word. These successive triplets eventually control the sequence of amino acid in protein molecule that is to be synthesized in the cell. Note the figure 3 to 6 that the top standard of DNA reading from left to right as the genetic code CAGACTT, with the triplets being separated from one another by the arrows. As well follows this genetic code through figure 3 to 7 is figure 3.8. We see these three respective triplets are responsible for the successive placement of the three amino acid proline, serine, and glutamic acid in a newly formed molecule of protein.
Figure 3 to 7. Combination of the ribosome nucleates with a strand of DNA to form a molecule of RNA, then carries the genetic code from the gene to the cytoplasm. The RNA polymerase enzyme moves along the DNA strand and builds the RNA molecule. Figure 3 to 8. A portion of an RNA molecule showing three RNA codes CCG, UCU, and GAA that control attachment of the three amino acid proline and serine and glutamic acid respectively to the growing RNA chain is achieved through the intermediary of another type of nucleic acid, RNA, the function of which is controlled by DNA of the nucleic. Thus, as shown in figure 3 to 7, the code is transferred to RNA in a process called transcription. The RNA in turn diffuses from the nucleic through nuclear pores into the cytoplasmic compartment, where it controls protein synthesis. RNA is synthesized in the nucleic form, a DNA template. During RNA synthesis, the two strands of DNA separate temporarily. One of these strands is used as a template for synthesis of an RNA molecule. The code triplet is the DNA result in the formation of complementary code triplets called codons in the RNA. These codons in turn will control the sequence of amino acids in a protein to be synthesized in the cell cytoplasm. Building blocks of RNA. The basic building blocks of RNA are almost the same as those of DNA, except for two differences. First, the sugar deoxyribose is not used in RNA formation. It is placed in its place is another sugar of slightly different composure, ribose, which contains an extra hydrogen ion appended to the ribose ring structure. Second, thiamine is replaced by another pyrimidine uracil. Formation of RNA nucleotides. The basic building blocks of RNA from RNA nucleotides, exactly as described previously for DNA synthesis. Here again, four separate nucleotides are used to form RNA. These nucleotides contain the bases adenine, guanine, cytosine, and uracil. Note that these bases are the same as in DNA, except the uracil in RNA replaces the thymine in DNA. Activation of RNA nucleotides. The next step in the synthesis of RNA is activation of RNA nucleotides by an enzyme, RNA polymerase. This activation occurs by adding two extra phosphate radicals to each nucleotide to form triphosphates by the two RNA nucleotides to the far right during RNA chain formation. These last two phosphates are combined with the nucleotide by high energy phosphate bonds derived from ATP in the cell. The result of this activation process is that large quantities of ATP energy are made available to each of the nucleotides. The energy is used to promote chemical reactions that add each new RNA nucleotide at the end of the developing RNA chain. RNA chain assembly form activative nucleotides using the DNA strand as a template. As shown in figure 327, assembly of RNA is accomplished under the influence of an enzyme. RNA polymerase. This large protein enzyme has many functions. Proteins necessarily form formation of RNA as follows. 1. In the DNA strand immediately ahead of gene to be transferred to a
polymerase has an appropriate promoter. The RNA polymerase is appropriate compartmentally structured and recognizes this promoter and becomes attached to it, which is the essential step for initiating the formation of RNA. After the RNA polymerase attaches to the polymer, the polymerase causes the unwinding of about 2 turns of the DNA helix and separation of the unwound proteins of the two strands. The polymerase then moves along the DNA strand temporarily stand unwinding and separating the two DNA strands at each stage of its movement. As it moves along at each stage, it adds a new activated nucleotide to the end of the newly forming RNA chain through the following steps. First, it calls the hydrogen bond of the form of between the end of the base of DNA strands of the and the basis of an RNA molecules in the nucleoplasm. Time. Then, one at a time, RNA polymerase breaks to the 3-phosphate reaction away from each of energy. From the broken high energy phosphate of remaining phosphate on the nucleotide, which
the transport vesicles instantly fuse with Golgi apparatus and empty their contained substances into the vesicular space of the Golgi apparatus. Here, additional carbohydrate moieties are added to the secretion. Also, an important function of the Golgi apparatus is to compact the endoplasmic reticulum secretions into highly concentrated pockets. As the secretion passes through the outer layer of the Golgi apparatus, the compaction and process proceed. Finally, both small and large vesicles continually break away from the Golgi apparatus, carrying them the compact secretory substances and diffusions throughout the cell. The following example provides an idea of the timing of these processes. When a granular cell is bathed in amino acid, newly formed protein molecules can be detected in the granular endoplasmic reticulum within 3 to 5 minutes. Within 20 minutes, newly formed proteins are already present in the Golgi apparatus, and within 1 or 2 hours, the proteins are secreted from the surface of the cell wall. Types of vesicles formed in the Golgi apparatus: secretory vesicles and lysosomes. In a highly secretory cell, the vesicles formed by the Golgi apparatus are mainly secretory vesicles containing proteins that are secreted through the surface of the cell membrane. These secretory vesicles are diffused to the secretory vesicle through the surface membrane. These secretory vesicles first diffuse to the cell membrane and then fuse with it and empty their substances to the excretion by mechanism called exocytosis. Exocytosis is most cases in simulated by entry of calcium ions into the cell. Calcium ions interact with the vesicular membrane and cause its function with the cell membrane, followed by exocytosis, opening of the membranes of outer surface and excretion of its contents outside the cell of some vesicles. However, the destined of the intercellular use are still for intercellular use of intracellular vessels to replenish cellular membrane. Some intracellular vesicles formed by the Golgi apparatus fuse with the cell membrane or with the membranes of intercellular structures such as the mitochondria and even the endoplasmic reticulum. This fusion increases the expansion of these membranes and replenishes the membrane as they are used up. For example, the cell membrane loses much of its substance every time it forms a phagocytic and pinocytic vesicle, and the vascular membrane of the Golgi apparatus continually replenish the cell membrane. In summary, the membranous system of the endoplasmic reticulum and Golgi apparatus are highly metabolic and capable of forming new intracellular structures and secretory substances to be extruded from the cell. The mitochondria extract energy from nutrients. The primary substance from which cells extract energy are foods that react chemical with oxygen, carbohydrates, fats, and proteins. In human body, essentially all carbohydrates are converted into glucose by the digestive tract and liver before they reach the other cells in the body. Similarly, proteins are converted into amino acids, that fat are converted into fatty acids. Shown in oxygen and other foodstuff, glucose, fatty acid, and amino acid all entering the cell. Inside the cell, they react chemically with oxygen under the influence of enzymes that control the reaction of the channels. The energy releases in the proper direction. The details of all these digestive and metabolic functions are provided in chapter 63 and 73. Briefly, almost all these oxidative reactions occur inside the mitochondria, and the energy that is released is used to form the high energy compound ATP. Then ATP, not the original food, is used throughout the cell to energize most all the subsequent intracellular metabolic reactions. Functional characteristics of androcyte triphosphate. ATP is a nucleotide composed of the following: the nitrogen-based adenine, the pentose sugar ribose, and their phosphate radicals. The last two phosphate radicals are connected with the remainder of the molecular by high-energy phosphate bonds, which are represented in the formula shown by the symbol. Under the physical and chemical conditions of the body, each of these high-energy bonds contains about 12,000 calories of energy per mole in ATP, which is many times greater than the energy stored in the average chemical bond, thus giving rise to the term high energy bond. Furthermore, the high energy phosphate bond is very labile so that it can be split instantly on demand whenever energy is required to promote other intracellular reactions. When ATP releases its energy, a phosphoric acid radical is split away and androgen diphosphate ADP is formed. This released energy is used to energize many of the cell's other functions, such as synthesis of substances with muscular contractions to constitute the cellular ATP as it used up energy derived from the cellular nutrients caused ADP and phosphoric acid to combine to form new ATP, and the entire process is repeated over and over. For those reactions, ATP has been called the energy currency of the cell because it can be spent and reformed continually, having a turnover time of nearly for only a few minutes. Chemical process in the formation of ATP role in mitochondria. On entry into the cell, glucose is converted into enzyme in the cytoplasm into pyruvic acid, a process called glycolysis. A small amount of ADP is changed into ATP by energy released during the conversion of this amount that contains less than 5% of the overall energy metabolism of the cell. About 95% of the cell's ATP formation occurs in the mitochondria. The pyruvic acid derived from carbohydrate, fatty acids, and other lipids and amino acids from protein is eventually converted into the compound acetyl coenzyme CoA in the matrix of mitochondria. This substance in turn is further dissolved for the purpose of extracting its energy by another series of energy is in the mitochondrial matrix, undergoing dissolution in a sequence of chemical reaction called the citric acid cycle or Krebs cycle. These chemical reactions are so important that they are explained in detail in chapter 68. In this citric acid cycle, acetyl CoA is split into its component parts, hydrogen atoms and carbon dioxide diffuses out of the mitochondria and eventually out of the cell. Finally, it is secreted from the body through the lungs. The hydrogen atoms, conversely, are highly reactive. They combine the oxygen that has also diffused into mitochondria. This combination releases a tremendous amount of energy, which is used by mitochondria to convert large amounts of ADP to ATP. The process of these reactions are complex, requiring the participation of many protein enzymes that are integral part of mitochondrial membranous slips that protrude into the mitochondrial matrix. The initial event is the removal of electrons from the hydrogen atom, thus converting into a hydrogen ion. The terminal event is the combination of hydrogen ions with oxygen to form water and release a large amount of energy to globular proteins that protrude like knobs from the membranes of the mitochondrial sleeves. These proteins are called ATP synthesis. Finally, enzymes ATP synthesis used in the energy form of hydrogen ions to convert ADP to ATP. The newly formed ATP is transported out of the mitochondria into all parts of the cell, cytoplasm and nuclear plasm, where it is energized multiple cell functions. This overall process of formation of ATP is called the chaomiosmic mechanism of ATP formation. The chemical or physical details of this chemical mechanism are presented in chapter 68, and many of the detailed metabolic functions of ATP in the body are discussed in chapter 68 and through 72.